Good evening, everyone. A very warm welcome to Syngenta's Gellets Hill Research Centre for the very first Arable Horizons lecture. My name's Carl Schneider. I'm the editor of Farmers Weekly and the editorial director in the UK of Farmers Weekly's parent company, ProAgrica. And I'm going to be your host for what promises to be a very exciting evening. The Arable Horizons lectures are based on a really simple idea. First, to identify the areas of science and technology that we think are likely to have the biggest impact on the way we farm crops in the UK in the next 10 to 15 years. And then for each of those topics, to find a leading researcher working in that field to paint us a picture of what farming could be like in that time frame based on what's happening now in the lab. I want to thank Syngenta for buying into that vision and for making it possible for us to turn it into a reality by sponsoring the entire series. Now, tonight's lecture will be delivered to an invited audience of farmers and others from the world of agriculture here in the Elements building at Gellets Hill. I think it's an appropriate place for us to kick off this series. It's Syngenta's biggest research centre in the world, and I'm told that that impressive glass house you can see out the window is the biggest research glasshouse of its kind in Europe. As well as those of you here in the room tonight, we have an audience online watching the live stream on Farmers Weekly's YouTube channel. And the whole event is being captured on video, and it'll be available at some point after the event on the Farmers Weekly website. So tonight's topic is plant breeding and genetics. And we're very fortunate to have for our speaker, Dr. Alison Bentley, Director of Genetics and Breeding at NIAB, the internationally renowned plant research centre based in Cambridge. Now, originally from Australia, Dr. Bentley is an agricultural science graduate from the University of Sydney. She's made her name for herself studying the way that the environment changes the way that different cereal varieties perform, and in the application of genetics in the breeding of high-yielding, climate-resilient cereals. At NIAB, she leads a 50-strong team doing groundbreaking work in plant research, applied plant research, and she's also chair of the UK cereals and breeding community known as Monogram. So this is how tonight's going to go. In a moment, I'll ask Dr. Bentley to come up and talk for just under an hour. Then we're going to break for coffee, and then we'll come back for half an hour of questions and discussion. And then to wrap up, we have Dr. Andrew Crossthwaite, who I'm told is literally speeding here as we speak after his plane was delayed from Basel, who's going to talk about how Syngenta is using some of the techniques that Dr. Bentley will talk about to develop exciting new products. We thought it was fitting for a series of events that are all about science and technology to exploit technology to make the events more interactive. So we're going to use a web-based tool called Slido, which allows you in the audience here, and also those of you watching on the live stream, to take part in polls and to ask questions. So if you want to participate, and I'd urge it, certainly everyone here in the room to make sure you do this, can you please now open your browser on your smartphone, if you're watching at home, on your computer or on your tablet, and go to www.slido.com. And when you get there, you'll see a screen that asks you to enter a code. There's a hashtag, and next to that hashtag, if you type in 2093, that should be all you need to do to get in. So I said later on we're going to use this in the Q&A session, and I think Dr. Bentley wants to use it a little bit during her talk. But just to show how it works, let's try it live now. And I've got my fingers very firmly crossed, having done live television about technology. I know how things can go wrong when you try and do things live. So let's try and do a live poll. And for our first question, we're going to try and find out a bit more about the audience we have tonight here in the room and those watching online. So a very simple question. What is your main occupation? Can you tap the appropriate response for yourself? Well, that's interesting. It's mostly other. So I suspect that's not quite as enlightening as I hoped it was going to be about our audience. Perhaps we should have put some other options on the screen. Okay, anyway, we've tested the technology, it works, so that's a good sign for the rest of the evening. So without any further ado, Can you please join me in giving a very warm welcome to our main speaker, Dr. Alison Bentley.
Hello and welcome to this talk. And I thought I would start the talk by telling you a little bit about my background and what seems to be an almost lifelong love of crop science. So as Carl mentioned, I grew up in Australia, which is a country very reliant on agricultural production. But growing up as a teenager on the outskirts of Sydney, I wasn't really acutely aware of this until I did my first experiment, which I've shown a figure from here. And in this experiment, our teacher asked myself and my classmates, he gave us a bag of potato tubers and asked us to grow these in plots on our school farm and apply different treatments to each of these plots, different water, different nutrients, and different weed control techniques. And at the end of term, we got the fun task of digging up the potatoes to see what had happened. And I'll always remember the light that came on in my head when we collated the results of this experiment, that I finally saw the point of studying biology, of understanding why plants grow, interact with each other in the environment. And that's what I hope to do tonight, to shine some light on genetics and breeding, and the importance that they may have for the future of farming. So I'm delighted to be here to give the first of these Farmers Weekly Arable Horizons talks here at Syngenta. And I like to think I've come quite a way from the potato plots of my teenage years. And as Carl said, I now lead the genetics and breeding work at NIAB. And this is a fantastic role to have because it means I'm able to work with a group of scientists who are really passionate about crop genetic improvement. And tonight I'm going to share with you some of the reasons why I think now is the most exciting time to be involved in or engaged with this field of crop breeding and genetics. And the reason for this is really for the first time we suddenly have all of these tools and technologies at our fingertips, which really have the potential to revolutionize plant breeding and by extension farming. So I'm really going to focus on these tools and technologies and the excitement that we feel about them and give you some examples of how we're using these technologies and how they can potentially be applied for the improvement of agriculture. And of course, I'm very interested in what you have to say about these issues, so please feel free to tweet me or use the Arable Horizons hashtag, or we'll also have these online polls which Carl mentioned during the talk and later during the discussion. So when I started planning for this talk, I thought about what are the targets that we hear about when we talk about crop genetic improvement? What are the things that people are interested in and talking about? And obviously, this is just a snapshot. This is my initial feelings on this topic. So we want new disease resistances to reduce the inputs we apply to our crops. It would be great if we could have cereal crops that produce their own nitrogen supply. Can we manipulate photosynthesis, this amazing biochemical way that plants convert light, carbon dioxide and water into energy and into food? And then also bigger picture questions like resilience of farming systems and sustainability, which will mean we'll be able to produce food and supply food in the future. And obviously high yield, this is a constant pressure to produce crops with higher yields for better return on investment, and greater use of the available land space for agriculture. And I put up a picture here of Norman Borlaug, who is the most famous of all wheat breeders and is known as the father of the Green Revolution. So Norman Borlaug introduced height-reducing genes into wheat, and by doing this, he created this boost in yield from better allocation of resources. And I think it's really interesting that even in the 1970s, Norman Borlaug saw that it was science and technology which were going to give us the tools that we needed to be able to increase and sustain our food supplies. So as I said, I'm really going to be talking about these tools and technologies, the methods by which we target each of these things that we want to do. We want to drive yield, drive sustainability. What are the tools that we have available? And I'm only going to talk about a handful of these. I could obviously talk for an hour on each of them, but then we'd be here all night. So I've brought this down to 5 areas that I'm going to talk about, from genome sequencing, diversity breeding, genetic modification, which is a very topical area, gene editing, which is an emerging technology, and then talk briefly about some of the breeding methods that are beginning to become available. And I think before I do that, I want to remind everyone of the reason that we would use any of these tools and technologies. And it comes down to the fundamental objective of breeding, which is to drive genetic gain. And this genetic gain is what you see here in this graph, and you can see yields increasing over time. And it's this increase in yields that we call genetic gain. And this is what breeders do. This is the data from yields in official testing trials over time. So you can see the impact of breeding, increasing the yields of our varieties. And breeders have a number of ways of increasing genetic gain, and really it's a numbers business, and I promise this is the only formula I'll use tonight. So we can quantify this genetic gain, this rate of increase of the yield potential of our crops, using a number of factors. And we can increase the selection intensity that we apply in breeding. So we can select fewer individuals from larger populations to increase the probability that those individuals are the superior individuals. We can increase the selection accuracy, so how accurately we can identify those superior individuals. We can increase genetic variance, so we're drawing from a wider pool of genetic variation. And on the bottom, we can reduce the time it physically takes to produce a variety. So if we can halve the time that it takes to produce a variety, we can double this response to selection. We can double the rate of genetic gain. So now I'll come on to my first topic, which is genome sequencing. And for those of you who are farmers, this may seem like something which is a long way away from farming, and may also seem like something that's a long way away from breeding. But I hope to show you that genome sequencing is really a foundation. It's an enabling technology. It's something that we need to be able to make genetic improvements to crops. And what is a genome? A genome is the genetic composition of an organism, whether it be a human, an animal, or a plant. So these are the DNA codes, the instruction manuals for an organism. And the reason we talk about genome sequencing so much is that until very recently, many of our major crop species didn't have a genome sequence. So essentially, we're not able to understand the genetic instructions which are controlling the things that we want to select for and that we want to improve in these major crops. And I'll also point out wheat here. So wheat has a very large genome, which is almost 6 times the size of the human genome. So we're dealing with something that's very complex, but we really need to access this information to allow us to genetically improve crops. And the fundamental reason why we want to know what the sequence of individuals are, what the genetic makeup of an individual is so we can attach it to a phenotype, to something that we see. And this really allows us to be able to select individuals more accurately, which you'll remember was part of that equation that I showed. So if we're able to associate something that we see with something that we know is underlying it as its genetic basis, then we're able to track that through a breeding process, and we're able to accurately select for the thing that we want to select for. I'll also talk about how we can use DNA sequencing to revolutionize our understanding of pathogens. And this is work done both at NIAB and the John Innes Centre in Norwich. And it's the pathogen you see on the screen, I'm sure many of you will recognize as yellow rust, which is a debilitating disease of UK cereal crops. So at NIAB, the UK Cereal Pathogen Virulence Survey started about 50 years ago in 1967, and it was initiated due to an epidemic of yellow rust on a very popular variety which had been susceptible to yellow rust before. And what this survey established was studies of new virulences of these yellow rust pathogens. So how the survey worked traditionally was that farmers from around the UK would send in an infected leaf sample from their field. The spores from this leaf sample, sent in an envelope, would then be multiplied and purified on a plant, and then used to infect a panel of wheat lines with known virulences, so known reactions to yellow rust. And this allowed the generation of phenotypic data, assessment of the range of varieties that these isolates of yellow rust were able to infect. And this process, in partnership with the John Innes Center, was revolutionized with the addition of sequencing. So if you add sequencing to this step, you still receive a sample in the post, but instead of putting it onto a set of plants, you now put that material into a sequencing machine, which is able to tell you the genetic code of both the pathogen and the plant that it's infecting. And what this does, it reduces the time drastically and means you can go from 25 samples a year, which is not particularly high throughput, anywhere up to 1,000 samples. And you now get genetic data on the pathogen that you're interested in and on the host. So this technique, known as field pathogenomics, is a great example of how you can combine a traditional pathology approach with sequencing and really access a much greater range of information. So if we look at the traditional pathology approach, this is a very rigorous approach. It tells us which wheat varieties are susceptible to yellow rust, and it gives us early warnings of changes in the profile of virulences that these yellow rust pathogens have. If we incorporate the sequence data, so we add on another layer of information, we're now able to answer a large number of questions. So if we have an isolate from the north and an isolate from the south, how related are they? Did they come in at the same time? Are they from the same population? Where did any new virulences, new races emerge from? What are the new risks? And are there any new associations between particular varieties going down with particular races of the pathogen? And I think the best example of this is the work that Diane Saunders at JIC did in the pilot study for this project, where she looked across 20 years of yellow rust isolates from the UK CPVS collection. And when she ran the sequencing procedure on these isolates, she found a very relatively small amount of variation within the UK pathogen population. Yet post the emergence of the warrior race, you can see the ability of this sequencing approach to identify this huge explosion in the virulences and the diversity of this pathogen. So this is something that was seen on farms, and it's something that we're able to detect using the sequencing technology. And this has also allowed for the development of a tool which can be used for real-time identification, classification of these rust pathogens. And this is a potential use both to breeders in terms of identifying new varieties and relationships with new races of the pathogen. It can be used to make variety choices. So literally the data comes off the sequencer and is available via this tool, which can be used to help decision making. So I think talking about diversity of a pathogen leads very nicely into diversity breeding. And this is a very broad subject which NIAB has been working on for the past 10 or so years. So all of our crops have evolved and been domesticated from ancestral forms, many of them which you wouldn't recognize as being the ancestors of crops. So here is teosinte, which led to the domestication of maize, which is shown here, a very different phenotypic look to the ear here. And here are some of the relatives of wheat. So we have huge amounts of variation. And here's some of the variation present in ancestors of domesticated potatoes. And the reason why we're interested in this diversity, which came from the ancestors of our domesticated crops, is that bread wheat, which is the example that we work on at NIAB, shows very little genetic variation. So the bread wheat that's grown in a farmer's field has a very narrow genetic base. And this picture is from a video we made a few years ago showing the science behind the processes that we're using to overcome this. So the problem with limited genetic variation is this what plant breeders rely on. So they need to be able to make crosses between things that vary for different traits. So if you don't have variation, you can't make this genetic gain that we see is vital to driving forward yields. So we essentially see that this limited genetic variation is a brick wall. And our question at NIAB is, how do we break down this brick wall? What can we do? And what we do essentially is time travel. So we go back to the very start of the process which formed wheat. So I mentioned before that wheat has a huge genome, and wheat also has a very complex genome. because, in fact, it's a single genome that combines the genomes of 3 ancestral grass species. And this is an amazing example of nature's genetic engineering, if you like, of putting together multiple grass genomes and creating something which goes on to feed the world. So the first of these chance encounters in nature occurred about 100,000 years ago. And this brought together in the wild grasslands of the Middle East 2 grass species, which are shown here. And by chance, these grass species, even though they had different genomes, different genetic compositions, came together. And this coming together produced wild emmer wheat. And wild emmer wheat has been domesticated to domesticated forms of emmer, which we see have health benefits and are quite fashionable types of grains. And these were also domesticated to pasta wheat, so the wheats that we make pasta or durum products out of. And then if that wasn't unlikely enough to have occurred, a second chance event in nature again happened much more recently, so 10,000 years ago. And this brought in an additional genome from a wild goat grass. And this second hybridization happened only 1 or 2 times. So a third of the genome of wheat, which grows in the field, is contributed from only 1 or 2 of these goat grass lines, which were growing in the Middle East and came together with this wild emmer wheat. So what do we know about goat grass? Basically, we know that goat grass contributes a third of the genetic information that is present in wheat. We also know that this happened very rarely, so we really have what we call a genetic bottleneck. So we're unable to induce new variation on the D genome. But obviously the addition of this goat grass made wheat what it is today. It gave it a selective advantage, so the early farmers were able to select these individuals which had spontaneously come together in nature because they had some favorable characteristics from this goat grass. And we know that wheat was domesticated in this region here to the south of the Caspian Sea, and we also know that this goat grass ancestor occurs in nature across a very wide geographic range, so all the way across the Tibetan Plateau. And our question at NIAB was, can we go back in time? Can we take these wild, diverse goat grasses and recreate this event, this evolutionary event which gave us the wheat we know today? And by doing that, when we're taking this geographic variation, will this incorporate new favorable genetic variation which we can use for breeding? And this is a picture of me quite a long time ago doing just that. So in the Middle East, searching for these wild grasses. And when I did this, I was looking for novel sources of disease resistance, because you can imagine wild grasses growing in natural habitats, having lots of different biotic and abiotic interactions in the environment. And in places like Turkey, you see these goat grasses, which are shown here. They really are growing as weeds among our domesticated cereals. So at NIAB, we go through this process of recreating this evolutionary event, and we call this resynthesis. And we call it resynthesis because we're remaking or resynthesizing an event that occurred in nature. So if you remember that wild emmer wheat was domesticated to durum or pasta wheat, we take this as the starting point. So we're only going back 10,000 years. We take this durum wheat and we pollinate it with these goat grass accessions. And very, very occasionally, this is a very technically demanding process, very occasionally you'll get something that produces a seed. But unfortunately, this seed only contains 1 copy of each of the genomes of the 3 species underlying wheat. So this is very unstable at this stage. So the wheat plant essentially tries to get rid of it. It tries to abort that seed, and that's the time when we step in, we rescue the embryo, we put it onto a medium which provides the nutrients that a seed would usually provide to the embryo. And you can see here it starts to produce roots and shoot tissue. We then use a chemical which artificially doubles the number of chromosomes present to form something which is stable. And this is called a synthetic wheat. This is a resynthesized copy of modern wheat. And the fantastic thing about this synthetic wheat is because its genome is identical to the wheat which would grow in the field, it's now fully crossable, and it's a vehicle for transferring this variation from these wild goat grasses into elite wheat. And that's exactly what we do as the next step in this process. So here you can see the original pasta wheat that we used in this crossing. You can see here, this is the primary synthetic. So this is the result of that cross between the durum wheat and the goat grass. It produces this. We then cross it with an elite UK variety. And you can see some of the reasons why we do this in the derivatives, which are shown here. So we have ear types which are far superior to the elite UK variety that we make the cross into. And this is what we're looking for. We're looking for novel variation that increases things we're interested in: yield, disease resistance, biomass. But it's worth saying that this diversity breeding approach is really like looking for a needle in a haystack. So we make hundreds of these crosses between these resynthesized wheats and modern elite wheats. And we look at them in the fields and interrogate them phenotypically to look for any of these favorable combinations. But it's very much like searching for a needle in a very, very large haystack. And a lot of the variation that we introduce is not favorable variation. So a lot of the material doesn't progress through the process. There's also the potential to use these wild relatives of crops as sources of novel disease resistances. And here I have a slide from one of our collaborators, Brand de Wolf, at the John Innes Center. And this is an idealized diagram of how you could source resistance genes from multiple wild grass species and then go through the process of incorporating them all into a single genetic background to produce a plant with novel resistances. But the problem between doing our diversity breeding, which I've explained, and doing this very targeted approach is that we need to know where in the haystack we're searching for these resistance genes. And one of the things we're doing at the moment is trying to find the money to be able to sequence all of these wild relatives, this collection, so we're able to better understand where we should be searching for these resistance genes so we can have a much more targeted approach to produce disease-resistant wheat. So next I'll talk about genetic modification, which is quite an emotive subject, and we'll stop during this section for one of the interactive polls. So how is genetic modification different from breeding? The approach which I've explained to you about diversity breeding was very much about let's throw in as much genetic variation as we can, and then let's look and see if any of it's good. Genetic modification, as I'm going to explain it here, is a much more targeted approach. So here we're looking to specifically target a gene of interest. And that is a very big difference. We're looking at very targeted modification of a gene or a set of genes in a genome. And how do we do this? How does this process happen, this genetic modification? And it can happen in one of two ways. The first, which is shown here on the left, is to use a bacteria called Agrobacterium. And this bacteria, we can insert the desired genes into something called a plasmid, which is a small circular piece of DNA which we can manipulate to insert the gene that we're interested in. We then co-cultivate this bacteria with the cells that we want to transform, and the plasmid incorporates this desired DNA into the host tissue. The second approach, which is more of a brute force approach, uses a particle gun to shoot particles which are covered in the DNA that we want to incorporate. It shoots them by bombardment into the plant cells, and transformation happens in that way. And it's worth pointing out that this Agrobacterium tumefaciens bacteria is a plant pathogen. So it's something that occurs in nature. It's really nature's genetic engineer. And what it does as a pathogen is it infects through the soil, through wounds in the roots, and it hijacks, it uses this ability to transfer DNA into the host cell, to hijack the cell essentially, to tell the plant signals to provide nutrients to the pathogen. And these result in these galls which are formed as a symptom of this disease. So when we use this Agrobacterium for genetic modification, the pathogenic part of it is disabled. We're simply using its ability to naturally genetically engineer its host and transfer some of its genetic information, which we've substituted for the gene that we're interested in, into the host cell. And I've got a picture here of sweet potato, which is a really interesting example, because one of the concerns about eating genetically modified food is that you're eating all of this bacterial DNA from this Agrobacterium tumefaciens. And that's seen as something that's not very positive. But I'd like to do a poll of how many people in the audience eat sweet potato or have eaten sweet potato in the past. So there's definitely a few owning up to it. So sweet potato, a recent study showed, contains significant amounts of this Agrobacterium tumefaciens DNA. And all varieties of sweet potato carry the DNA of this bacteria. And this is the result of genetic engineering in nature. So none of the ancestors of sweet potato have this Agrobacterium tumefaciens DNA in them. This event, which naturally genetically engineered sweet potato, happened post the domestication and has obviously been selected on unconsciously by people who have taken forward favorable versions of this sweet potato. So now I'm going to use the online poll to ask you about genetic modification. And I asked my group recently if you could transform or genetically modify one gene or one trait in a crop, what would it be? And so you can either use the hashtag GM targets to let me know the type of targets that you would have. Then we can probably discuss them later. But I think it's interesting to show some of the things that my group came up with when I asked this question. So first, we've got genes that trigger a rhizobial association, which is related to this ability of wheat or a cereal to fix nitrogen. So this is obviously a target that I'm going to talk about later. And then we have some things about grain quality, so wheat that's safe for celiacs, or generally targeting grain nutrition. Then we have some bigger picture things like drought resistance in wheat, bolting resistance in sugar beets to produce winter varieties, pod shatter in oilseed rape, so these are practical targets that we could achieve. Then we have some more interesting ones, allelopathy in wheat, so wheat that produced chemicals or signals that suppressed weed growth, so you wouldn't need herbicides. Herbicide resistance for Africa and for particular crops. And then this is my favorite, I think, that we can produce high-yielding blackgrass that we can eat. So I think this would really be a landmark example of the use of genetic modification. So now I'd like to give some examples of how we're using genetic modification at NIAB and how some of our collaborators and friends are using genetic modification to make you aware of the kind of activities and the uses of this type of technology. And this is a slide from Anna Gordon, who works at NIAB, and it shows the reproductive tissue of wheat being infected by a fungal pathogen, Claviceps purpurea. So Claviceps causes the disease ergot, which produces these black sclerotia in the ear. So it's an interaction between the fungus and the reproductive tissue of the plant. And these ergots are toxic to humans and livestock. And so altogether, this is a disease that we would like to understand more about the interaction between the pathogen and the host as a means to control it. And an interesting observation that Anna made was that when you look at the relationship between plant height and the size of ergots, you find that shorter plants have less ergots. And this got Anna thinking, is this just a physical characteristic? We see this with other diseases like Fusarium head blight. Is it just a case that this plant, which was shorter than all the other plants, escaped the infection of the ergot pathogen? Is it something physical, or is it something about the hormones and the chemicals in a plant that make it short or tall? And to ask this question, is it just a chance effect of the architecture of the plant, or is it something to do with the way the plant signals and develops this height? What we really needed to be able to do was to turn off this growth hormone, which is called gibberellic acid, which controls the height of a plant. We wanted to turn off this hormone, and we wanted to turn it off just in the ovule tissue, so only in the reproductive part of the plant. And Anna wanted to ask the question, how does turning off this growth hormone in a single part of the plant affect the ergot production. So to do this, she did it via genetic modification. She took a gene called GA2 oxidase from runner bean, and this is a gene which suppresses the production of this hormone, and then she put that into a wheat plant. And you can see here in the figure, which is with the genetically modified plants parts stained in blue, that the result of this was specific expression of this GA oxidase, this suppressor of this growth hormone, was localized to this reproductive tissue, and that's where we wanted it to be localized. So this technique allows us to target a specific gene to a specific part of the plant. And this is early days still in this study, but you can see that in all of the cases, the sclerotia weight in the lines with this suppression of this hormone, gibberellic acid, had significantly lower sclerotia weight. So by manipulating the signaling, by cutting off the expression of this single gene, we were able to show that this is a mechanism to increase the resistance of wheat to this pathogen. So this is a very simple example where we've asked about the relationship between a pathogen and the host, and some of the mechanisms which are involved in that interaction. And we're able to use genetic modification to switch things off in a very specific tissue to understand more about that relationship. I'll come now on to some of the bigger picture challenges that people would like genetic modification to address. And one of the big ones is the use of nitrogen. So nitrogen accounts for up to 70% of wheat's carbon footprint. So this is obviously a huge part of agricultural production, and also a huge part of the environmental effects of crop production. And this is a figure that scares me a little bit. So we can see these are projections these are based on FAO projections of fertilizer use. And you can see these increase across the board by 2050. I think most striking in places like South Asia, this huge growth in this huge forecast projection in fertilizer use is really something that we need to take stock of, and we need to start thinking about the solutions for this. And some of these may lie in crop genetics and breeding. So quite often people say it would be great if you didn't have to apply any fertilizers to a crop. So one of the ways you could potentially do this is to produce cereal crops which made their own nitrogen, therefore reducing the need to apply any nitrogen to the crop. And this is exactly what's happening, and I present here some slides, again from the John Innes Centre, from a project which is trying to do just this, trying to engineer this nitrogen symbiosis in maize. And just to remind you, there are two different types of root symbioses, so interactions between roots and microorganisms. The first one is the one we know quite a lot about this interaction between rhizobia bacteria and the roots of legumes to produce these nodules which supply nitrogen back to the plant. So this, at the moment, is restricted to leguminous crops, leguminous plants. Then we have a second type of root interaction, which is an interaction with fungi called mycorrhizae, and these are inside the root tissue. This relationship is possible with about 90% of all land plants, so most plants can make these mycorrhizal associations, and these mycorrhizal associations help with the uptake of phosphorus. So this is the framework within which Doreen and the ENSA project are working. So basically, in order to make a cereal which made these rhizobial associations, these nodules on its roots to supply nitrogen back, all you would have to do is switch the current signaling pathway which leads to these mycorrhizal associations to nodulation. And this might sound like a fairly easy thing to do. They currently share this kind of first part of this complex signaling process, so all we'd have to do was manipulate it. So at this point, it switched to this side and produced these nodules. So doing that is actually a very ambitious task, even with sequencing, even with very efficient genetic modification, because manipulating any of these things in isolation is very complicated to do. To be able to modify them or change them and then stick them all together in a biological process is a very difficult ask. So this is what we need to get to in order to produce nitrogen-fixing cereals. So I think this is a high-risk, high-reward approach. If it works, it would be fantastic to be able to modify something that already exists in the cereals, to modify that to produce nitrogen. At NIAB, we take a much more modest approach to addressing this nitrogen challenge. And our interest at NIAB in this project called Syntron is to look at incremental gains in plant nitrogen use efficiency. So actually, can we break down a plant and the ways it uses nitrogen and modify each of those component parts? This will allow us to understand what's the effect of changing one thing on nitrogen use efficiency, and then perhaps we can put them all together and design a plant which has the optimal additive use of nitrogen. So we do this by modifying the angle of the leaves, because branching in crops, in all plant species, is regulated by nitrogen. The remobilization, so the way that plants remobilize nitrogen from their leaves into their grain or their oil, and increasing plant biomass. So this is a much more incremental approach to improving nitrogen use efficiency. So I think if nitrogen-fixing cereals are the high-risk, high-reward of molecular biology, we can still make progress, which is much more incremental, which allows us to understand how plants use nitrogen and to build up a picture of what the optimal nitrogen use type looks like. But what about yield? Genetic modification for yield, obviously, that would be a very big win to be able to produce a plant by genetic modification which had higher yield. And hopefully you've seen in the press that Rothamsted have recently applied to DEFRA to grow trials, field trials of genetically modified wheat, which has been shown in a glass house to have increased yield. And what's underlying this? So what the researchers at Rothamsted and the University of Essex have done is they've overexpressed an enzyme which is part of the photosynthesis process. And just to remind you that photosynthesis is the biochemical machinery of a plant which takes in sunlight, carbon dioxide and water and turns these into sugars which support plant growth and yield. And photosynthesis, I'm afraid, like all of the processes that I've already described, is very complicated. So we have multiple things going on at multiple times and lots of enzymes and reactions happening within a chlorophyll, a chloroplast. And you can see what the researchers in this case have done. They've overexpressed just one of the enzymes which is part of this reaction, this biochemical reaction. And the hypothesis that they're testing is that increasing the activity of this SBPase will increase the activity of rubisco, which is one of the rate-limiting enzymes of photosynthesis. And by doing this, you can drive yield by making photosynthesis essentially more efficient to produce more of these sugars, to produce more yield. And I'll put this, so obviously the next step for this, which has shown very promising results in the glasshouse, is to trial it in the field. And obviously we await the DEFRA permission for the trial and the results of it. But I think it's important to say now that the ability for us to grow and test these materials which are made using genetic modification, whether they be for a single gene, an enzyme, and a complex pathway, it's very important for us to have the ability to test these in the field, to look at the actual effect of manipulating specific parts of a plant and how they can affect yield and growth and the interaction with the environment. So now I'll talk about gene editing as an extension of genetic modification. And gene editing is a very exciting technology which really has the potential to revolutionize what we do with plants. And editing techniques all work in the same way. They use a nuclease, which is essentially a molecular pair of scissors, to make a very specific cut at a specific position in the desired gene. And the reason we hear more about gene editing now is because of CRISPR. So there are other techniques for gene editing available, zinc fingers and talons, but these are very complicated to use. They rely on proteins and the 3D structure of proteins and are very difficult to actually use. This is in contrast to CRISPR, which essentially relies on a DNA sequence, an RNA sequence, so one of these strings of genetic information. But the take-home message is they're all essentially a pair of scissors which allows you to cut at a very specific position in a gene. So Emma Wellington, who leads the transformation at NIAB, provided me with this slide about how does CRISPR work. And it looks quite complicated. And basically what we have is we have designed something which will recognize the gene that we want to cut, and then we attach to it the Cas9, which is the nuclease. This is the scissors. So we've got a target sequence we want to cut, and we put the scissors onto it. And then this is able to recognize the DNA sequence. So this you see here is the binding of that guide and that pair of scissors onto the gene that we want to make the cut in. And then this pair of scissors makes a cut. And the real magic of gene editing is that what we're actually doing by cutting a gene in a specific place is triggering mutations. So the DNA will try and repair itself. It will jam the ends of those cut fragments back together. And what this does is causes a genetic stop signal. So this is a perfect way to knock out a gene, so to remove the function of a gene by cutting it and inducing this stop. And I talk about this as well. This is the basic design of this, the thing that we are inserting. And the important thing, which I'll talk about later as well, is that we're able to separate out all the parts of this. So we're able to separate the bit that we want to add in, which is here, to tell the scissors where to cut, the scissors and the selection. So we still need to be able to select for edited plants in tissue culture. And we have a selectable marker here, but we're able to select away from that at the end of the process. And I'll tell you why that's significant in a minute. So we know that gene editing can be used to stop genes. So can we use it to knock out things that we don't want? And I'm using another example from work that's ongoing at NIAB from one of our PhD students, Arable. And this work recognizes gluten, which we obviously hear quite a lot about, and it's present in most edible products. So wheat grain gluten is a polymer. It's made up of multiple components. We have the glutenins, which are the structural components, the gliadins, which are responsible for the viscosity, and then the ratio of the two gives us the bread dough quality for baking. But people with celiac diseases have a very complex interaction between their immune systems and between gliadins, so this component of the wheat gluten polymer. So what Arable is asking is, can we use gene editing to provide new solutions for people with celiac disease? So the first solution is to eat a gluten-free diet. So gluten's in most of our foods, so we would have to exclude cereals from our diets. The other option is, can we produce cereals without these immunogenic properties? So can we knock out the bits of gliadin which are causing this immunogenic reaction, this negative relationship between a host's immune cells and the gluten that they're eating? So to do this via conventional breeding would take a very long time and may not be a feasible approach. So therefore, using gene editing, this technology by which we can stop something, we can knock something out, is a very attractive option. And this is what Arable is testing at the moment with CRISPR, with this gene editing technology. And essentially, she's trying to knock out a very specific region of these gliadin epitopes to make wheat that's not toxic to people with celiac disease. And again, this work is only just starting, but the early results are very promising, and they show that by using this CRISPR technique, Arable is able to change the profile of the gliadin components of the grain. So this work is really exciting in our ability to knock out things that we don't want while maintaining the rest of the characteristics that we want. And obviously in the case of alpha-gliadins, we know that the gluten to gliadin ratio is a determinant of bread dough quality. So we need to understand a lot more about this, because we may actually knock out these gliadins and then not be able to make bread anymore, which is obviously not an ideal solution either. So in terms of gene editing being close to market, so there are already plants, crops on the market which were created using this gene editing approach. This includes a waxy hybrid maize, browning-resistant button mushrooms for processing, and improved storage potatoes. And then if you look in the literature and what's around, there's a lot more coming. So some of these were made with the older technology, and now we have these CRISPR gene-edited plants coming through. And a big question, which I haven't dwelled on with the genetic modification, but which is really relevant to gene editing, is what will the regulation be? So in this approach, we're using a much more targeted approach. We're essentially inserting a pair of scissors into a gene. And in the US, this hasn't been regulated as a genetic modification, and this is the same in Argentina and Sweden. And in Europe, this is a really big question, which will ultimately determine the use of this technology for crop genetic improvement. And we've been awaiting a verdict on the regulation of gene-edited plants for quite some time. I think it's also important to ask, how will public perception differ to genetic modification, which has obviously been a very sensitive topic in Europe. And we need to think about how we talk about this gene editing and the potential of it and what it actually does to a plant. And this is a great slide that I've taken from Hugh Jones at IBIS, which shows you this kind of framework of regulation. So if we have a conventional genetic modification, like I've described, we've inserted some DNA into that. So we've inserted a piece of DNA into a host genome. This is a genetically modified organism and is regulated as such in the EU. If we use mutation breeding, so if we take a bag of seeds and apply very high dose radiation to it, or some mutagenizing chemical to it, we induce multiple changes in the host genome. And these are untargeted. We don't know where these are. And this mutation breeding has been used since the 1930s to develop food crops. So we induce variation by making lots of modifications to a host genome. And although we're making modifications to a genome, we don't really know what we're doing. And this is not considered to be a genetically modified organism in the EU. So then if we use this gene editing approach, and we go through this step, which I showed you the design of the CRISPR-Cas9, if we're able to separate out the pair of scissors, the original guide, and the selectable marker, which allows us to select the individuals which have been edited, what we end up with after this process of removing all the bits of machinery that we use to make the edit, we end up with a host genome with a very targeted edit. So a genome that looks like this with a single edit in it. And for me, all of the examples I've presented are things which are very complex. So when I see a simple answer, I grab hold of it. And to my mind, in this instance, there's no way of knowing how this edit, how this mutation was made. So in that respect, it's very difficult to see how you can regulate something that you can't see. And so I think it's very important that we show that this gene editing technology produces something which is not very dissimilar from what we would get from inducing mutations via mutagenesis, via radiation or chemical means. But we await this outcome of this judgment on the regulation of genetically edited plants. And now finally, I'd like to talk very quickly about breeding methods. And breeding methods are really things that we can use now to make things quicker. And as I said at the start, if we can halve the time that it takes to produce a variety, we can double the genetic gain or the response to selection. And one of our collaborators at the University of Queensland, Lee Hickey, is doing this using a concept known as speed breeding. And this was taken from NASA's attempts to grow plants in space. And you basically saturate the plants with light. So you have almost 24-hour lights on a plant, which makes them photosynthesize, produce much quicker. And they've shown with spring wheat, this allows you to get through 6 generations a year, and you can screen the plants for rust, for diseases as you're going through this process, and make crosses at the same time. So these are types of practical things that we can use now to speed up the process of breeding. We also have F1 hybrids, which probably deserve a mention at a talk about crop genetics and breeding. So F1 hybrids, depending on where you make your money, are a very attractive option because you have to buy seed every year. And the reason that you buy seed is to exploit hybrid vigor. And hybrid vigor is what you get, so you cross 2 parents together, and you get progeny here that outperform either of the 2 parents used to make them. And the level of this outperformance is really important to the success of hybrids. So at the moment, we don't see these gains, this hybrid vigor for things like yield to an extent that justifies necessarily the expense of buying the hybrid seed. But this is really a technology that's been burning for a long time, but we now have some of these genetic tools which may allow us to make bigger and faster gains in hybrid breeding. And I'll put in another technology which is a long way off, and it's called apomixis. So the reason with hybrid seed, the reason why you need to buy seed again the next season is if you planted the resulting seed, it would be segregating in the next generation. So it's a mix of 2 parents, and in the next generation, you would get a completely mixed field because of the way the genetics behind it works. And here I've put a picture of an idealized apomixis situation, which is asexual reproduction by seed. So in this case, you would be able to make an F1 hybrid, and then that F1 hybrid would produce the same seed in the next generation. And this technology, this apomixis, is not yet available for any of our seed crops, but is really something that people are working on, and it has potential to change the way we buy or sell seed. So I'll come back now to finish about a reminder of why we use all of these tools or technologies, and that is our intention to drive genetic gain, so increase the genetic potential of our varieties. We can do this by selecting harder, we can select more accurately, and a lot of the tools I've talked about allow us to be able to select individuals much more accurately. We can reduce the breeding cycle time, and we can increase the amount of genetic diversity that we can use for breeding. And of course, many challenges remain. I've touched on the regulatory issues around gene editing, and we know about the issues around genetic modification. I think it's interesting that recent work has shown, not working anymore, that 2/3 of the public would actually now back GM crops, and Brexit is a great opportunity potentially for us to assess our stance on the use of genetic modification in crops. And to summarize, I've talked about 5 of the tools and technologies which have the potential to revolutionize the way we breed crops, the way we understand crops, and potentially the way that farming works in the future. And I hope you'll agree that these tools and technologies are an exciting part of the way we understand crops and our potential to improve them. And finally, I'd like to acknowledge the people who've contributed slides to this presentation. So people from NIAB, the John Innes Centre, CEH in Edinburgh, the University of Queensland and IBIS. And a lot of our funding comes from the UK government, from the BBSRC, and the UK CPVS, which I've talked about, is sponsored by AHDB and AFA. So thank you very much for your attention, and I look forward to answering questions during the discussion. Thank you.
Well, thank you very much, Dr. Bentley. It certainly wet my lips thinking about the opportunities that some of these techniques could could give us. So we're now going to take a break, and we're going to go through there to get some coffee. We're going to take a 10-minute break, so it's now exactly 7 o'clock, perfect timing. So we'll come back in here at 7:10 for a Q&A. Now, I said that the technology, the Slido technology, allows us to answer polls, but also ask questions. So if during those 10 minutes you want to ask a question in the second part, go to Slido and tap on the tab at the top. There's two. One says polls and one says questions. If you tap on the questions, and then there's a box there for you to type in your question. And in the second part, I'll be sitting up here. We're going to change the set, so we're going to have a couple of comfy chairs for Alison and I to sit on, and we're going to go through some of those questions and answer them and have a bit of a discussion. So I don't feel you just have to ask questions. If people have something to contribute from the floor, we'll deal with that. Obviously, Dr. Bentley's focus is on the tools and the future and the research. We've got, I forgot his surname, Rob Jackson from Syngenta. So if we've got any questions from the audience about sort of the here and now and some of the traits that are being developed in varieties now, then he's on hand to answer some of those questions too. So don't forget, ask your questions on Slido, go and get the coffee, and I'll see you back here in 10 minutes.